Good evening all of you. So how are you all? I hope you all are fine. So this is, all of you must be aware that tomorrow is the NEET and it's a big day, not only for a student but for all of us, right? And in fact for you also. Tomorrow you will see what kind of paper is asked and that will set a tone that how you should start preparing. So tomorrow is a big day for all of us. So okay, good evening. Yes, good evening Lucky. Good evening Chandrika. So my voice is clear? Yes, Nirant Sai. Sunday is NEET. So tomorrow will be the NEET. Yes. So yes, the class is also, today is the last class for botany. So let's make this class memorable so that you also have a very good memory of this subject. And I hope you have learned a lot of things in this entire journey. We have started with the basics and then gradually we are trying to understand complex thing. Yes, good evening. So my voice is clear as I can see here. Good evening Charan, good evening Faz, Chandrika, Lakshmi, Krishna, Mallikarjun, Krishna. Yes, good evening. So now just I can see that some of you are regularly connected in the class. Some of you are regularly responding. And I can just, I haven't seen your face, but many names are there whom I can now easily recognize because they are the first one to answer and they will answer every question. So I will like to congratulate all those students who have attended all the class without fail and you have learned a lot from here, right? So without losing much of a time, let's start with let's complete the topic which we have started in the last class, and in the end we can also have some conversation about subject in general and how you will approach NEET in botany. So in the last class, if you can recall, we have talked about the cell cycle, right? In which we have seen that the cell cycle is the sequence of event in which the cell divide. Sequence of event in which the cell divide, and here it is divided into two parts. One part is the preparatory phase, right, in which the cell do the preparation for the division, and another part is the division phase, which is also called M phase. But this happen in the eukaryotic cell. In prokaryotic cell, of course, we have seen first why cell divide. So these are simple things. Now we have started here what are the different types of division. So here amitosis in prokaryotic, mitosis and meiosis in eukaryotic. We have already discussed it. Then we have seen what is amitosis and this part. So this I will just once again I will repeat this part because last class I have seen some of the comment where you have told me to repeat this once again, right? So I am again repeating it, and it is very important also. If question from cell cycle is asked, then question from this interphase is very important because it's a little confusing also at times for students. So see, interphase is the longest. Interphase is a very long phase because it consists of more than 90% of the duration of cell cycle. It is divided into G1 phase, S phase and G2 phase. G1 phase is called gap phase 1. S phase is called synthesis phase. G2 phase is called gap phase 2. Now G1 phase, it is the longest phase. In G1 phase what happens? Protein and RNA synthesis take place. Whereas in S phase what happens? Synthesis, so DNA synthesis and the centrosome synthesis. Whereas in G2 phase protein and RNA synthesis take place. Overall, we have also seen cell cycle in human cell is 24 hours and yeast it is 90 minutes. Then we have seen some of the exercise based on that, and then we have seen the different stages one by one. So the first stage is the prophase. So prophase we have gone through the prophase. So in prophase, we have seen that prophase is the longest phase, and here there are several parts of the cell which will disappear, and these include the nuclear membrane, Golgi complex, ER and nucleolus. See, previous year question is asked. And what was the question? Question was very clear. Question was that which of the following cell organelle will disappear during prophase? And options were there. Correct answer was all of the above, right? So these are the cell organelle which will disappear. Centrosome, it will move toward pole. See, these are the two poles present over here. And then this chromosome will condense, means chromatin will become chromosome. And finally, spindle fiber is formed. As you can see here, the spindle fiber is formed. See, the main purpose here is to divide the chromosome present in the cell into two halves. Now, for division, you need two pole, and spindle fiber is like a thread, right? It is the thread through which you will bind to the chromosome, and it will pull on one side and it will pull on other side. Understood? Two pole, through the thread they will bind with the chromosome and they will pull it into two opposite direction. Clear? So here it will happen. So first, prophase, this happen. Now then second, so in prophase what we have seen, all the cell organelles have disappeared. Now then comes the second phase, and that is called metaphase. Now in metaphase what will happen? In the metaphase, chromosome will get arranged onto the equatorial plate like this, right? It is called the metaphase plate. It is called the metaphase plate. Two centrosome are present on the two poles, right? These are the centrosome present. Now, this is the spindle fiber, spindle fiber, spindle fiber, spindle fiber, right? So let us understand this diagram a bit. All of you pay attention here. Now, here the chromosome are attached onto a plate, right? And this is called metaphase plate. This is called metaphase plate. Clear? So one of the characteristic of metaphase is chromosome are arranged. In school, what happened during the assembly? We all stand in line. In the same way, chromosome also stand in a line. Clear? Then two poles are there. So from both the pole, spindle fiber will be formed. Spindle fibers will be formed, right? And this is the pole is represented by what? Pole is basically the centrosome. Understood this? Now tell me one question. In the structure of chromosome we have studied it. The spindle fiber bind to which part of the chromosome? Question is there. Tell me. Spindle fiber binds to dash of chromosome. Tell me the answer. Tell me the answer of this, right? Let me see who can answer it. Very good, Chandrika, very good. Very good, Chandrika. Okay, so Chandrika, tell me, in centrosome what is present where it will form? Janu also have given right answer. Very good. Now tell me what is there in the centrosome. Sagar, correct. What is there in the centrosome in which the see, centrosome is not centromere. Remember, here this is centrosome. Here not spindle fiber is binding. Spindle fiber is binding here. Remember. Okay, 1 second. This part is what? This part is the centromere. right? What is present in the centromere? We have seen here, each centromere has what? Each centromere, very good, Faz has given answer, kinetochore, Reddy, kinetochore, very good, very good. So as I can see here, if you say this is the centromere, if this is the centromere, centromere will have two kinetochore. Understood? Each centromere has two kinetochore and spindle fiber will come and join to this kinetochore. Understood this? Here, this we will call it as, centromere. This we will call it as kinetochore. This we will call it as spindle fiber. Spindle fibers, right? So is this point clear? What is centromere? Centromere is present at the center of the chromosome. Inside the centromere is two kinetochore. Each kinetochore, the spindle fiber goes and attached. So if question is asked, spindle fiber get attached to which part of the chromosome? So either you will say centromere or more precisely you will say kinetochore. Is this point clear? Now, so here this is what will happen. Apart from this, in this stage, metaphase, chromosome is best visible. If you want to see the chromosome, which phase you will choose? You will choose metaphase. Why? Because it is maximum condensed. Clear this point? So there are few points. First thing, in the metaphase, chromosome is attached to the metaphasic plate. So this is one point you have to remember, attached to the metaphase plate. Second, the spindle fiber is attached to the centromere. Which part of centromere? Kinetochore. Then chromosome are best visible and it is maximum condensed. Here what will be the answer? Spindle fiber binds to. So I will say kinetochore. If I ask you question, let me see how many of you can answer it. Tell me, spindle fiber bind to kinetochore. Fine. If I ask you, each centromere has how many kinetochore? Question. Each centromere has how many kinetochore? Tell me the answer. Let me see. Okay, Chandrika, very good. Janu, very good. Venkata, good. Pagidela, correct. Very good. Vijay Lakshmi, Apex, Sagar, Misbah, Sweetie, very good. Ganpati. So all of you are correct. As we can see here, one centromere has two kinetochore. So as every day you all are responding very well. Okay, so somebody just asked me one question and because answer are coming very fast, it went away. So can you write answer? I could just read that, sir, what is the difference between, and after that I could not read. So just please write that question again, I will read it. So yes, second is the right answer here. Very good. So in the metaphase, chromosomes are present onto the metaphase plate. So till this point everything is clear. Now let's move on to the next phase. See here, and try to understand it. These are the two pole, these are the thread, and thread has bind with both sides. Now what will be the case? Now the thread will pull each other in opposite direction, right? Pulling the thread. So when the thread will be pulled, what will happen? This, remember, this is pulled on this side and this is pulled on this side. Won't the centromere will split, right? So the next stage there will be a splitting of centromere. That happen when? That happen in the anaphase. So now we are going to see anaphase. See, in anaphase what will happen? Just let's understand this part a little. If I am telling that this is the case and it is pulling on this side and pulling on this side, what will happen next? You will see that the chromosome breaks, the centromere breaks, and one start moving toward this pole and another will start moving toward this pole, isn't it? Two chromatids are there, one chromatid will go on this pole, another chromatid will go on this pole. And why it happened? Because here the centromere is present and here there is a splitting of centromere, right? So I am going to write this in clearly. So this point you understood. So now let's write. First thing which happened here during anaphase is splitting of centromere. Splitting of centromere. When there is a splitting of centromere, will sister chromatids separate? Yes, sister chromatids separate. We have already seen what are sister chromatid. So these two are sister chromatids. Sister chromatids, right? These two are sister chromatid. Now you can see sister chromatid separated, sister chromatid separated, right? Now here splitting of centromere and sister chromatid separated. So this is the characteristic of anaphase. So how will anaphase look like? Let's see this diagram. Here, see, isn't it happening? Here the centromere is present and this, here the centrosome is present here, the centrosome, this is the spindle fiber, and this will pull the chromosome on to opposite side, right? So this is happening in anaphase. Now, one more important point you see. Here chromosome number is 2, but here what is the number of chromosome? 1, 2, 3, 4. So in anaphase, remember, chromosome doubles. Chromosome doubles. Remember, so if I ask you in which phase chromosome number get double, so you will say in anaphase. And finally in the telophase, new nuclear membrane is formed, right? So here, once this part is done, right? Once this part is done, then what you will see that, see, if this is a cell, so one of the chromatid will come on this side and another chromatid will come on this side, isn't it? They have separated. Now what will happen? Here a nuclear membrane will be formed. Nuclear membrane will be formed. Understood? See, nuclear membrane disappear in which phase? Disappear in which phase? Prophase. Nuclear membrane reappear in which phase? So you will say telophase. In telophase, all the cell organelle, remember, Telophase is exactly opposite to prophase. Telophase is exactly opposite to prophase. Clear? Means what happened in prophase? Nuclear membrane disappear. Here nuclear membrane reappear. Nucleolus disappear. Here reappear. Golgi reappear. ER reappear. Right? There you see chromatin become chromosome. Here what we will see? Chromosome become chromatin. There what we see? Spindle fiber appear. Here spindle fiber disappear. So everything is opposite. So we can simply say that it is opposite to prophase. Why we say? Because first point, here what happened? Nuclear membrane, nucleolus, Golgi complex or Golgi bodies, both are the same thing. Then Golgi body, ER, they all reappear. Then what happened? Second, in prophase, chromatin become chromosome. Here what happened? Chromosome become chromatin. Chromosome become chromatin, right? Third point, important. What happened here? Chromosome become chromatin. Third point, spindle fiber disappear. Spindle fiber disappear. Spindle fiber disappear. So overall here what we see? Now the karyokinesis is completed. Karyokinesis consists of how many steps? 4 steps. Prophase, metaphase, anaphase, telophase. Right? Is it clear? So now the karyokinesis is completed. So I guess that most of you must have understood these 4 steps. So as always, I will not let you go this way. I will ask question, right? I have this habit of asking question in class also. I will teach one topic and then ask question, so that half of those who are sleeping or half sleep, they will wake up in answering, and also those who are alert, they will revise also, right? So let's do some question and we will have the idea that how much you have understood. I can see more than 170 students are connected here, right? So all of you, if you are enjoying, hit the like button also, right? So let me see how many likes you are. This is the last class. Okay, so let's start with a question. So this question I will ask in a fill in the blanks format, right? You have to just give answer in one word. You have to give answer in one word. So let's see. Very simple question. Karyokinesis has dash steps. How many? You have to write the number, right? You have to write the number. Yes, I can see likes are increasing. Very good. So you have to write the number here. So first question, very good. Faz has given first answer. I will try to take the name of those who answer first. Chandrika, Ritika, Sagar, right? Janu, Venkata, Yashwant, Reddy, Krish, very good. Misbah, Yadav, Ganpati. Yes, it has 4 steps, right? Prophase, metaphase, anaphase, telophase, right? Now, so this was easy one. Now we are getting into complex one. Out of these 4 steps, you have to just write the name, right? So first, I know you are all alert and you will answer very quickly. Centromere split in. Tell me, in which phase? Centromere split in which phase? I am waiting. Who will answer first? I will take the name. Very good, Manoj. You are telling all the steps. Now answer this. Very good. Khalil again has given right answer. Anaphase. Janu, correct. Anaphase. Chandrika, Anaphase. Nagar, Anaphase. Reddy, Lucky, Misbah, Venkat, Manoj, Sriram, Sagar. Very good. So you all have understood it, right? It takes place in anaphase. Very good. Now third question. Third question. Listen it very carefully. Nuclear membrane. Nuclear membrane reappears in. Nuclear membrane reappears in. When? Reappears in. Very good. Khalil again has given right answer. Telophase. Very good. Khalil has given answer telophase. Narsimha. Okay. Chandrika, Ganpati, Praveen, Venkata. Very good. Reddy, Nikhil. Very good. So you have given telophase. So very good. So telophase. Telophase. Right? So it will be telophase. Then fourth question. Tell me, chromatin becomes chromosome in which phase? Chromatin start becoming chromosome in which phase? Very good. Again, Khalil has given first answer. Very good. So it is prophase. Chandrika. Shloki. Right. Very good. Venkata. Reddy. Sagar. Reshma. Okay, I am, chats are coming very fast, so I am not able to read all the names. But I am very happy that some of you are answering every time correctly. Right. So all of you who are answering correct. So it is prophase. It will be prophase. Very good. Prophase. Now let's see the fifth one. Chromosome best visible in. Best visible in which phase? Chromosome best visible in which phase? Very good, Khalil. Chandrika got the answer. Right. Vijay Kumari. Very good. Reddy. So chromosome is best visible in metaphase because it is maximum condensed in metaphase. Now next and last question from this section. The last phase of karyokinesis. The last phase of karyokinesis. Which is the last phase of karyokinesis? Very good. Very good, Khalil. I have given right answer. Jyoti, Pinky, Sagar, Praveen, Narsimha, Litesh, Chandrika. Very good. Lasya, Vijay, Rani, Yashwant. Very good. So all of you are giving right answer. The last phase of karyokinesis is telophase. So now I guess that things are very much clear about karyokinesis. So first we have seen interphase, which consists of G1, S phase and G2 phase. Then we have seen the M phase, in which we have seen karyokinesis, which consists of 4 stages, right? Prophase, metaphase, anaphase, telophase. We have seen what happened in which stage, right? Now, if I ask you one more question, I missed it, so answer it first. Seventh question, last question. In which stage number of chromosome doubles? I hope it is visible here. In which stage number of chromosome get doubles? Tell me the answer. Number of chromosome. Again Khalil has given right answer. So I should give star to Khalil because he has answered maximum number of question first. So, and all those who have given correct answer, these stars are for you. You all are doing very good, right? Maintain this discipline for 2 years and nobody can stop you from becoming a topper, right? So all the energy which I am seeing in all of you, just maintain the same energy in the classroom also, and nobody will stop you from getting into the medical college, and nobody can stop you from becoming a topper, right? It is the discipline, self-discipline which will decide, right? So now let's move on to the next part. So after karyokinesis, the next stage is cytokinesis, right? Next stage will be cytokinesis. So cytokinesis means cyto. So here if we see, cyto means cell. Cyto means here cytoplasm, right? So once nucleus is divided, then division of cytoplasm is called cytokinesis. Now here identify which cell is this. See, the boundary is circular. So whenever the boundary is circular, you will say it as the animal cell. And whenever the boundary is a square, cube or rectangle, then you will say it as the plant cell. This is the way to identify. Now if you see the animal cell, here you can easily see there is a furrow formation, right? There is a furrow formation like this. Means the membrane is getting squeezed inside, right? So remember, in animal, the 2 points you have to remember here. First is furrow formation, and second is the membrane is moving from outside to inside. Outside to inside is called centripetal or centrifugal. Physics. Tell me, is it centripetal or centrifugal? So it is called centripetal, right? Now here in case of plant cell, because plant cell is cell wall, it is very hard layer, so this cannot come inside, right? So there is no furrow formation. Instead there is a cell plate formation. What is formed? A cell plate is formed, and this cell plate grow from center to outward. When there is a center to outward, it is called opposite of centripetal. It will be called centrifugal, right? So simple 2 point of difference you have to remember. Furrow formation in animal cell, cell plate formation in plant cell, right? And here the furrow formation will be in centripetal direction, and here it will be the centrifugal direction. Is this point clear to all? So that is it about the cytokinesis. So overall we have understood this entire stage. Now this is mitosis. So here the number of chromosome will remain the same. Remember here, this is one cell, right? And from one cell you get two cells, in which first the nucleus divided and then the cytoplasm divided. So this is indicating both karyokinesis and cytokinesis. Now, here if the number of chromosome is 2n, so here also the number of chromosome will be 2n, here also the number of chromosome is 2n. So, in mitosis, number of chromosome remain the same. Number of chromosome remain the same. Is it clear? Now, if let's say in one division, one cell become two cell, right? So on this basis, there is one very interesting formula or interesting question which is asked. Let's say, if I tell you that one cell become two cell, let's say in 20 minutes. One cell become two cell in 20 minutes. So, tell me, after 60 minutes, how many cells will be formed? Question. It's a little tricky question. Think about it and then answer. What I am asking, listen carefully. And then in the end, I will first try by yourself and then I will give you a very simple formula, right? Okay, one question is asked that, sir, why is the cell is having 2n? See, here, first understand, this is 2n. Now, you are seeing that it is divided into two parts, so it should be n and n. No, why it is 2n, 2n? Because you can remember here during S phase, what has happened? Isn't DNA replication happened? So DNA replication, what it will do? It will double the DNA content and then it is halving. Basically understand, it is first multiplying by 2, then dividing by 2. Means one replication and one division. So won't it be equal? See, first replication into 2 and then division divided by 2. So won't it be both 2 and 2n? Right? So yes, that was the question. Now I am getting the answer. So my question I will repeat again. See, one cell become two cell in 20 minutes. So after 60 minutes, how many cell I will get? So very simple. See, in 20 minutes, one become 2. In 40 minutes, 2 become 4. And in 60 minutes, 4 become 8. 4 become 8. But see, this is one simple, if simple question like this is asked, then we can answer it very easily, right? But now I am, so here as I said, I will tell you a formula through which you can easily answer. So the formula is first division time. Now let's say here division time, what I have given you earlier question, I said 20 minutes. Now let's say division time is t, right? And then I give you total time. Total time is capital T, right? Total time capital T, division time small t. Then number of generation. Number of generation will be capital T divided by small t. That will be the number of generation. That is n, right? Understand this point. If I tell you division time is small t, total time is capital T, right? So in the last question what I have told you, division time was 20 minutes, right? Total time 60 minutes. So number of generation how much? Answer quickly. Number of generation how much? 60 divided by 20. Isn't it 3? Then number of cell, final. Number of cell will be equal to simple formula 2 to the power n. No need to do that calculation every step, writing the time and counting it. Simple formula 2 to the power n. So 2 to the power 3, 8. Isn't it simple? See, last question I have given you a very simple number. That's why you might have done in this way. But many times you will get complex questions. So let's do one question based on this, and then let's understand you are able to do it or not. So this formula, I guess all of you must have noted, right? Capital T by small t and then 2 to the power n. So here, let's say if I ask you division time, I will give you simple number. Division time is 1 minute, right? Division time is 1 minute. Then tell me how many cell in 4 minutes. Simple question. How many cell in 4 minutes if the division time of a cell is 1 minute? So, and the initial number of cell is also 1 cell. So here simple question. Let me see who can answer first. Chandrika is telling 4. Okay, this time Chandrika has given first answer. Venkata is telling 16. Sagar 16, Ritika. Okay. Okay, Faz also have given. So this time Faz is a little late. Okay, 16. So see, again, same formula we will use. Capital T divided by small t, 4. And number of cell is 2 to the power n. So 2 to the power 4. Simple. So it is 16, right? So just by seeing the number, you don't have to fear, right? Everything is very simple. Just it need 4, 5 or 10 question in botany or overall in bio. These number based questions are fixed. If you practice 10 question, you will always get the same question again and again and again, right? So overall we have done this chapter. So I guess all of you must have enjoyed it. So if you want me, then I can again give you some more questions.
No, sir, if you are done, I will come.
Okay, so these were the questions, and so thank you all of you, right? So this was the last class, and I hope that all of you must have enjoyed the class. It was a very wonderful experience for me, and many of you were very much regular in the class, right? And this is how we teach regularly in the classroom. Many things we could not do, like reading NCERT, asking individual questions, but all those things we do in the classroom. And all of you are very punctual, very prompt here in answering. Continue this. And as I have told you earlier, if you be so much dedicated and disciplined, then nobody can stop you from getting into medical college and getting a top rank, right? So thank you all of you. So
thank you, Kunal sir, and hello, this is Dr. Gandhi. I'm one of the executive deans of Sri Chaitanya. And anyways, as it is the last class for your bridge course today, I hope, because you might have seen me while introducing all the teachers in the first session, as well as introducing your course in the first session. I hope you guys have got some good amount of knowledge, as well as I'm seeing so many people. I'm very sure that every one of you got enlightened and at least got some base for your preparation. అంటే మీరు ఏదైతే అనుకున్నారో దేనికోసం అయితే మీరు ప్రిపేర్ అవడానికి వచ్చారో దానికోసం ఒక ప్రాపర్ బేస్ మీకు this eminent faculty who have taught you might have given you, and that's the reason Sri Chaitanya wanted to give you the best of the best team, so that your base becomes, or foundation becomes very strong. And I am very, very happy that whenever I see the live sessions, I see your energy. Your energy is fantastic. You guys are very prompt in responding, answering, and the amount of energy you guys throw in the chat box is fantastic. And keep having the same energy. Happy and stay happy, stay healthy. And tomorrow we have a NEET examination, and I wish all the NEET aspirants who are going, on behalf of you, your parents also, best wishes for them. I wish everybody fulfill their dreams, because in 2027 at this time you all will be taking the exam, and at this point of time we will be with you, working with you and working for you, and you will be in the same nerves of writing the exam. And anyways, all the very best for the bright future. We want to, the motive of these sessions, or the motive of this program is to build a strong foundation, and you are the doctor in making. Before the doctor in making, these all steps and these are inputs by your teachers will surely help you. Please carry and follow each one of them, and this habit of answering, this habit of being regularly interactive in the classroom, wherever you join and learn from, is very, very important to grow you and grow your knowledge, as well as make your learning process very active. Once again, thank you so much for being an amazing crowd, amazing learners, and we wish you all the very best from whole Sri Chaitanya family. Thank you so much. All the best.